Year 11s, we're going to be looking at genetic engineering, and specifically we're going to be looking at how this has been applied by humans either in industry or medicine or realistically in any field. Before we even consider this though, we need to consider this idea of artificial selection, because genetic engineering is basically exactly that. Artificial selection has been done by humans for thousands of years, tens of thousands of years in fact. When we look at our pets, our dogs particularly, we have bred dogs for specific traits. When we do something, when we breed an animal for a specific trait, we call that selective breeding. When we go through a process of selecting specific species based on traits that we enjoy or traits that we find desirable, that's artificial selection. And it's counterposed against natural selection. We are acting as the selective agents. We are the ones that are choosing which traits are acceptable and which traits are not desirable, which is why we end up with ridiculous sort of breeds like teacup pigs, where they have no real benefit to themselves naturally, and in fact it causes significant disease. This is one of the concerns with genetic engineering, and we'll get to that in a moment. But first we need to look at what actually artificial selection is. So if we take our example, so we have an apple tree. And the apple tree produces red apples. And that's what we're hoping for. That's what we're desiring. And we have another apple tree. And this apple tree produces some red apples, but it also produces some black apples, so some unhealthy, undesirable apples. What we would do with artificial selection is we would cross these together. We would breed those together, and our hope would be that in doing that, we end up only with trees that produce red apples. Now, we can get into the genetic analysis of all of that and how the likelihood of that, and that's not relevant for now. This is just how we artificially select organisms, or have done in the past. And the obvious reason for not doing a dog is because my drawing skills are not particularly good. But this is what we have done for an extraordinarily long period of time. And it doesn't matter what kind of food group or what kind of animal or what kind of plant you look at, we have done this. The majority of the plants that you have in your garden have been a product of artificial selection. It's just a very, very simple process, or at least artificial breeding. With genetic engineering though, it's a particular kind of artificial selection. Instead of hoping that we end up with mostly these red apples and not the black apples, not the unhealthy, nasty apples, we ensure that that's the case. Instead of going through this selective breeding process, we go through a process where we actually take the gene that we desire and insert it into our target organism. Now, it is literally limitless. If you have an organization or an industry or a medical practice that is going to be using living organisms, then the ability for genetic engineering to impact that is as big as your imagination can go. Anything that we could potentially engineer, any living organism that we could potentially engineer, is going to become an incredibly valuable resource. So there's this big, big push in genetic engineering at the moment. And it's got its concerns. But it is probably, or quite possibly, one of the largest sources of medical and industrial revolution in the next 20 to 30 years. It is that large. Now, you actually need to look at some engineering techniques. So there are a series of questions that you need to be able to answer. The table over on the next slide actually gives you a list of which kinds of techniques you're going to be using. So there are 4 there. There's plasmid insertion, which is a relatively old technique. There's CRISPR, there's TALEN, and there's one called zinc finger insertion. Now, I understand 2 of those. I haven't done a bit of research on those. The other 2, TALEN and zinc finger insertion, I have no idea. So you're going to need to go about and do that research. Answer the questions, seek feedback, develop your understanding. Then you're going to come back and you're going to educate the class on that. You'll see the instructions on that slide, on slide 100. If we go over now to slide 102, we're looking at the current benefits of genetic engineering. And so we need to look at what they actually are. It's too simple just to say that genetic engineering has its medical benefits. It obviously does. We can look at cancers and bone cancers, and certain types of gene therapy can help with other forms of cancer. Remember though that genetic engineering is limited in what it can do for individuals, because it is only able to affect organisms or affect people when it's done at the zygote level, when it's done pre-implantation. So this excludes any kind of natural reproduction. It excludes its sex-based reproduction. It is artificial, so we would have to use IVF. Now that's not necessarily a concern. It may or it not be based on your personal beliefs, but it is a limitation. And whenever we're thinking of limitations that are based around somebody's access to medicine, or access to technology, or access to money, we have to be concerned about the social impacts of that. And this is basically where genetic engineering is at. What are the social impacts of genetic engineering? The benefits are obvious. Medical, industrial. If we can make a yeast that's able to produce 200 liters of alcohol per hour, that's going to save breweries and winemakers an incredible amount of money. It's going to save them time, it's going to increase their productivity, it's going to have a huge impact. And in a country like or in a state like South Australia, where we have a significant amount of research and investment and money involved in the wine industry, it becomes incredibly important. But these benefits are broad. They are very broad, not just medical. So you must keep that in mind. The industrial benefits are equally important. So then we look at what the risks actually are. Now, genetic engineering is reasonably new. So there is that concern to start with. But we also think about those social risks, those social concerns. What about access to it? Who is going to access? Who's going to decide who does and who doesn't have access? Is it going to be based on who can pay? Is it going to be based on who actually needs it? Most countries don't have a system like ours, a Medicare system where the government pays for the majority of our medical care. So in a lot of countries like America and large parts of Europe and certainly the developing world, your access to medicine is largely based around how much you can afford. And if you can't afford treatments like this, generally speaking, gene therapy is hundreds of thousands of dollars per session. If you don't have $100,000, then you get to die of cancer. That's how simple it is. So what are your treatment options? What's actually available here? This relates to your SHE task directly. There are a whole bunch of other issues that they discuss there. But the big thing I want to focus on, because it relates directly to your assignment, is what are the social implications? What are the ethical implications about access to this kind of technology? It might not relate directly to your cancer research, but it does relate to just consistently thinking, what is this impact? How is this impacting here? And then lastly, we're just having this last focus on microbes in industry. We haven't had much of a chance to develop this. Now, microbes, these microorganisms, are incredibly important from an industrial purpose. There's nothing really to draw on the screen, on the whiteboard about this. This is really just a discussion here. So the more we understand about microorganisms, the more we understand about what we've learned so far, what they require for life, what they actually need for their survival, how we can treat them better, how we can improve their living conditions, how we can increase their productivity, is going to have a terrific impact on our actual ability to produce what we need to. Even down from the perspective that if we consider the ethical concerns around this, and we're using yeast, for example, to create alcohol, is it appropriate that we genetically engineer that yeast? Or is it not? Should we, shouldn't we? What are these discussions that we need to have? But again, microorganisms are used in a huge amount of different different industrial groups or industries. One of the biggest and more interesting, biggest in terms of development, is in fuel. Fossil fuels are obviously going to run out. They are finite, as we talked about in our carbon cycle discussion. They're finite. So then we need to have this discussion around what are we going to do next? One of the big things is biofuels, particularly algal biofuels, algal diesel or biodiesel. And we've actually managed to genetically engineer or produce algae, artificially select algae, that produce large amounts of this biofuel, of this biodiesel. And they're starting to trial this in things like airplanes. Not commercial planes, but in private planes now. And they're finding that they're doing more and more research and finding more and more out about these and manipulating these so that they have a terrific amount of variation to them. There's a video there that I'd really like you to watch about using different alternative energy sources and how algae particularly or bacteria are involved in this. So it's worth your looking at that.